Welcome back. I hope you attended a good breakout session and had a nice lunch. I am super excited for the next session. I'm going to introduce Dr. Rainer Surges, who is a longtime PAME friend. I think we've known you since the very beginning and has done all sorts of work and has a super important role right now as the chair of the ILAE SUDEP Task Force. For those of you who don't know ILAE, it's the International League Against Epilepsy. So Rainer is a trained adult neurologist with special qualifications in sleep medicine and seizure disorders. He's a professor of epileptology and chair of the department at the University of Bonn in Germany. His clinical research interests include autonomic function in people with epilepsy and pathomechanisms of sudden death in epilepsy, mobile health technologies for seizure detection, and epilepsy surgery. He's the current chair of the ILAE Task Force, as I just said, and has served for 10 years as the chair of Patient Safety Commission of the Germany Society of Epileptology. So welcome. We can't wait to hear what the task force has been up to.
Yeah, good afternoon. Dear family members and colleagues. So if you are asking yourself why there is only one country that has national guidelines on the use of seizure detection devices, you see the reason just in front of you, because obviously we tried to push it very hard because we believe that it's very important. Now, this work that I'm going to talk about has not been done on my own, obviously. We are international group of experts who have been nominated by the ILAE. So this is the most authoritative society in epilepsy disorders. So we have an impact with our work. And although it's going to be maybe a little bit dry, my talk, I will explain you why it's important, why we believe that we can make a difference with our work. So the group is composed of more than 20 members, well balanced, and this is very important. We also have child neurologists on board and experimental researchers. So we have the full spectrum of perspectives on mortality and SUDEP in epilepsy. So our mission obviously is that we want to contribute to the reduction of the incidence of sudden death in epilepsy. When you form an ILAE task force, you have to define objectives, and obviously we have very important ones. We want to identify priority areas in knowledge and practice related to SUDEP. We want to promote multicentric research, trials on SUDEP, prediction and prevention. We want to improve understanding, knowledge, and awareness of SUDEP among epileptologists, but not only among experts, but obviously also among general neurologists, pediatric neurologists, medical examiners, and general practitioners. And we want to promote the collaboration with key stakeholders and organizations, also with industry. So you have objectives, but obviously you also have to deliver something. At the end of our mandate, after 5 years, we have to present something. And we've defined this with respect to education. So we were aiming at planning scientific symposia, patient webinars. So these are low-hanging fruits. But we also had this idea of getting more complicated work to do by revisiting SUDEP definition, classification, and to write up a consensus statement on SUDEP counseling. So this is just to give you a flavor of the low-hanging fruits. So this is an advertisement of our webinars that you can even nowadays visit and consult on YouTube. So here's the website on ILAE, and you can click on it, and then you have the webinar as a YouTube video. So there's one for patients and caregivers. There's one for physicians as well. Okay, let's go to the big chunks, which are very difficult. For what reason? Because we try to describe something that is usually unobserved, the sudden death in epilepsy. And there's this recent definition that was coined almost 30 years ago and then recoined about 10 years ago. And this is the recent definition. Sudden unexpected death in epilepsy is a category of death in people with epilepsy occurring in the absence of known structural cause of death, and is most likely heterogeneous with regard to mechanisms and circumstances. And here you see already that this definition is so wide that it's difficult to improve it, because it has to fulfill many criteria. It has to fulfill, let's say, truth. So every case that we consider as a SUDEP case should be covered by this definition. But then also, if we are too precise in the definition, we run the risk of losing information in cases that might be due to SUDEP, but we don't know exactly. So this is something that is very difficult to redefine. And I'm going to explain you that we were fighting on 3 major changes in the definition, and we were fighting for each and every word. So why do we need SUDEP definition? Obviously, you heard about the assessment of SUDEP incidence, so how often does it occur per year in a given population. We want to monitor trends. Are we getting better in preventing SUDEP? We want to compare different studies, and obviously we want to advance research. This is the reason why we need to define it so properly. So there are different categories. One category is the definite SUDEP. So we believe that we are sure that this is SUDEP. And this is the definition. You probably heard it this morning already or yesterday. It's defined as a sudden, unexpected, witnessed or unwitnessed, non-traumatic and non-drowning death occurring in benign circumstances in an individual with epilepsy, with or without evidence for a seizure and excluding documented status epilepticus, in which post-mortem examination does not reveal a cause of death. So basically what you've read here is that we don't know exactly what SUDEP is, and we define what it should not be. So this is important to understand. And now here, this is what we had decided already, how we would improve this definition of definite SUDEP. So basically, we omit the unexpected. I will explain you why this is important. And why do we have to revisit definitions and classifications? Because other definitions and classifications undergo modifications during the years. For instance, there's a new definition of status epilepticus that was introduced like 5 years ago. And this older definition refers to this, so we had to change it. And this is how we changed it. We said, or we were discussing that we should exclude all death that is due to documented status in a sense of a convulsive status epilepticus lasting longer than 30 minutes. Now, why do we believe that eliminating the word or the term unexpected is important? Who has ever heard of the sudden, unexpected cardiac death? I haven't. So if it's sudden, it's usually unexpected. And like 15 years ago, there was this other term. This was unexplained. So the time given, like 15 years ago, the wording was changed from unexplained to unexpected, but we believe that unexpected, we just can omit it. And this is also for, in my opinion, a political reason, because this term, unexpected, emphasizes the unexpectedness, and this may fuel nihilism. That people believe, oh, I don't have to talk about SUDEP because it's unexpected. It's so rare that we don't have to talk about it. I think this is wrong. And this is also because, omitting the term unexpected also stresses that, SUDEP is partly preventable. We know this. We've learned this during the last 10, 15 years. So this was, for me, although it's, you might laugh about it, but for me this was one of the major advances, because we are going to call it sudden death in epilepsy, and the important thing is that we keep the acronym or the abbreviation SUDEP. So this was one, for me, major step. Now, what is the third big thing that I believe is super important? Now, one of my conflicts of interest is that I'm very much interested in everything that is heart, cardiac mechanisms, and so forth. And why am I saying this? Because we know that sudden cardiac death is not rare, that in 1 of 1,000 people per year in the general population, it occurs. And people with epilepsy, we know this during the last 10 years, have an almost 3-fold elevated risk of dying in sudden cardiac death. Meaning that if you assume that the incidence of SUDEP is 1 per 1,000, then sudden cardiac death risk is 3 times higher. So this is an important population that we need to address, and with this, let's say, a greater precision in explaining the overlap between sudden death in epilepsy and sudden cardiac death, this will raise awareness, and this will also allow and empower physicians as well as patients to improve cardiovascular health in their patients. And at the moment, we believe that this elevated risk of sudden cardiac death is due to anti-seizure medication. We know that if you take sodium channel blockers, everybody does almost, this alone increases already the risk of sudden cardiac death, even in the absence of a known cardiac disease. Then there are some epilepsy-related factors that we do not understand so much so far, but also the fact that in people with epilepsy, they have more frequently these typical risk factors of cardiovascular diseases, such as high blood pressure, elevated levels of cholesterol, and so forth. So why am I saying this? Because at the moment, the recent category of, let's say, contributing or competing cause of heart is only implicitly mentioned in the SUDEP definition as so-called SUDEP plus. And this means that there's a case that satisfies the definition of definite SUDEP, and there's a concomitant condition, for instance, a cardiac disease that is identified before or after the death, and that may have contributed or not to the death. So this is not very clear, obviously, on the one hand, and on the other hand, these cardiac problems, they are so prevalent that it needs to be reconsidered and mentioned. And why is that? Now, let's be honest, we all live in a bubble. So we live in the epilepsy SUDEP bubble. And do you know this saying, to a man with a hammer, everything looks like a nail? Well, I can explain to you that this is true. If I were a cardiologist and I come up with my definitions of this is sudden cardiac death and here and there, you will find out that the definitions, they resemble each other. So if I'm a cardiologist, I find a young person who is dead in bed, I would say, this is probably sudden cardiac death. If I'm an epileptologist and this person was known as having epilepsy, I would say this is SUDEP. The problem is that as an epileptologist and a neurologist, I will never dive into the cardiology literature. So we have to bring it to the epileptologists and neurologists. And this is the reason why I believe that we need to put it into the new classification and definition paper. And this is how we believe that there's a proper overlap of SUDEP and SUDEP Plus and what is called sudden arrhythmic death syndrome. I don't want to go into detail. I just want to tell you that this is all true. This exists and there are overlapping entities, and we have to define this precisely. And why is that? Because if you have this tonic-clonic seizure that results in primary apnea, so no breathing any longer, and then the heart stops beating. This is a very different mechanism of, the heart paces very, very fast, so fast that there's no blood that comes to the brain, and then people die. So if you want to target the mechanism and if you want to reduce the risk, you have to exactly define what you are targeting at. And this is the reason why we need to be so precise in the different definitions. Okay, we felt that this was too much to put this in. Oh, I've got 1 minute? Oh, wow. So, will I get a bonus, a jet lag bonus? So we felt that this is too much to put this all in one paper, so we decided to develop a second paper where we will propose a research framework where we integrate the challenges as well as the progress that we made during the last decade. What are the progresses? Obviously, we have novel technologies. We have made progress in data sharing. This is important for artificial intelligence approaches and so forth. We have identified leading mechanisms and cascade. This is the reason why we want to put this into a second paper where we define exactly the mechanisms, and we want to prompt researchers and clinicians to be more precise. What are they targeting at? If they want to plan, or if they plan, let's say, an interventional study, they want to find out how good wearable devices are in reducing SUDEP incidence, well, then they have to define that they are aiming at SUDEP cases which are due to this fatal cascade tonic-clonic seizure and then primary apnea and so forth, but they won't be able to target primary cardiac arrest because there is no tonic-clonic movement. So we have to be more precise. Okay, now I have to hurry up. So there's this third project. This is a consensus statement on SUDEP counseling. We know that the provision on information on SUDEP risk factors and how to mitigate the risk is still not part of standard epilepsy care in all regions and countries, although it's recommended. And so we have very ambitious people in the working group, in the task force, who've made a scoping review, and they're creating items for a Delphi process that Johan has already mentioned. So this is a method to get opinions and agreements on specific topics. Okay, I hope that I have you convinced that obviously it seems to be a very dry topic, but as an ILAE task force we can make a difference, and this hopefully has an impact on the awareness of SUDEP, that we want to support appropriate care for people with epilepsy, raise awareness and active management of cardiovascular health in people with epilepsy, and support the planning of better studies. Thank you very much.
That was great. Thank you so much. That was great. And maybe next year we can have
Hopefully.
more and partner more with ILAE, because it's awesome that you are championing this work, and it's exciting to see this issue being reframed in that way. So thanks so much. Okay, final plenary, come on up. Tom is moderating, right?
Oh wow, those lights are bright. Hello, it is an honor to be here, and I want to thank PAME for inviting me to be one of the moderators of this next panel. I'm Karen Utley, and I wear a few hats. I am co-founder and president of the International Foundation for CDKL5 Research. I'm the vice chair of the Rare Epilepsy Network, and I'm also a registered nurse. Let me see if I can get to... But this little lady you see right here, she is the reason I wear all of these hats. This is my daughter, Samantha. She is 18 years old, and she's living with CDKL5 deficiency disorder. Her diagnosis catapulted me into a life I never expected to have, but I have tried to rise to the occasion. A few years ago, I started attending PAME, and I was so moved and encouraged by this effort. I want to thank every parent in this room. You have lived through the worst possible loss, and yet you are here, and you are trying to help make sure that other parents don't have to live through the same thing. You are valued, you are appreciated, and you are making a difference. Last year, I heard discussions around other causes of mortality besides SUDEP, and I was encouraged, maybe even a little inspired. SUDEP is not common in the CDKL5 community. Most of the deaths are from known causes, but it's mortality just the same. I started thinking about possible opportunities to work collaboratively on prevention of all mortality, and I shared my ideas with some people, and REN, the Rare Epilepsy Network, decided it was worth exploring. And we reached out to Gardner, and she encouraged us, and we kicked off, sorry, I lost my place. We kicked off a working group, an exploratory working group. Allison Kukla and I are co-chairing this working group. And as you can see from this slide, REN is a group of over 150 rare epilepsies. It's an established collaborative network, and it was a great place to start. And I left that sentence off, so sorry for sounding a little broken. So our goal is to find a way to make a difference, and areas of opportunities we identified are also on this slide. If any of you are interested in this, please consider joining us. This is not exclusive to REN. But now for kind of why we're here, I'm excited to kick off this next plenary session around the State of SUDEP Innovation. And with that, I'm going to hand it over to Tom.
Thanks, Karen. I really appreciate all the work that you do for REN and our community. Just a brief comment. The first memory that I have thinking about innovation as it relates to SUDEP was a comment that my brother Mike made after his son Danny passed away. It was 2009, and technology existed in every facet of life. And I remember he said, how could there not have been something that alerted us to a seizure when Danny was sleeping? And so the premise to increase family knowledge and access to technology was in large part the reason that we started the Danny Did Foundation. And our session is going to cover several points of innovation within epilepsy. Researchers' understanding of the possible causes of SUDEP has grown over the last 2 decades with various preclinical models that now exist. We've heard about some of them here at this meeting. The treatment of epilepsy has also shifted over time, and the advent of precision medicine and targeted therapies, despite advancements about our knowledge of possible SUDEP causes, specific treatments for prevention are still lacking. So this session is going to explore the opportunities for growth within the field of SUDEP prevention from clinical and basic science approaches with a talk from Dr. Luis Lopez Santiago, innovations in gene therapy with a talk from Dr. Heather Mefford, a discussion about surgical options from Dr. Sandy Lamb, and finally, the use of epilepsy technology with remarks from Dr. Nathan Fountain. And at the end, we'll have time for some questions. So to move this ahead, it's my pleasure to introduce our co-moderator, Dr. Robin Whitney from McMaster Children's Hospital in Ontario, Canada. Welcome, Robin.
Thanks, Tom, and to Karen for moderating this session with me. It's my pleasure to introduce our first speaker, who is Dr. Luis Lopez Santiago, who is an associate research scientist from the University of Michigan in the Department of Pharmacology. He has over 25 years of experience doing patch clamp electrophysiology, and he's going to be speaking about how our knowledge from SUDEP animal models can be applied to trying to develop novel treatments to prevent SUDEP.
Hello, thank you for the invitation to talk. This is the first time that I'm in PAME, and I'm very motivated to keep doing basic science research because all the we have been learning today.
Yeah.
So all we know what SUDEP stands for, and we have some new definitions that I think are going to help us a lot to understand each other. Also, we know that numbers change depending on the study, but there are SUDEP occurs in approximately 1 to 5 in 1,000 people with epilepsy annually. And especially for Dravet syndrome, where mortality rate is a lot higher. And that is something that I will be talking a little bit more, Dravet syndrome, because it is the area that we are working more in the Laurie Eisen laboratory. So to understand SUDEP, it's very important to have some animal models because they can recapitulate various human pathologies, including mechanisms for epilepsy and SUDEP. And also, this allows us to have a control experiments to study these mechanisms. Most of these animal models are genetically modified organisms, and we can classify them into types. Most of the ones that we use are animals like rodents, like rats and mice. There are many of them. I just put a few examples here. One of them is to study the mouse autogenic seizures that is very well characterized. We have been learning a lot from them. Also the SCN1A, that is the Dravet syndrome model that we use in the laboratory. But there are many others for sodium channel alpha and beta subunits, calcium and potassium channels, and receptors. The second group is non-rodent. Those are human, not human primates like baboon and marmoset. These are not genetically modified, but they are very important because they have similar physiology than humans. And the newest model that we are working on is rabbit SCN1A that also models Dravet. Animal studies have helped us to understand some possible mechanisms of SUDEP. They allows us to understand how the brain interacts with heart and with lungs. There are multiple findings. It's not possible for me to talk about them today in a short talk, but I want to point at least 1 or 2 points of every one of them. So for respiratory mechanisms, we know the seizures induce respiratory arrest after generalized tonic seizures in mouse models. And they have helped us to understand the circuits and possible mechanisms for respiratory arrest. We can see here a picture or a drawing of the mouse brain where we can see circuits and brain regions that they are very similar than humans. And that is the way that we can understand these respiratory mechanisms. The new rabbit model that we are working on also is helping us to understand other things. We can see on the left, the wild-type rabbit, they have a normal breathing pattern, and with that is a normal respiratory and sinus arrhythmia present. On the other hand, the SCN1A heterozygote rabbit, they have altered respiratory rhythm, and with that, they have a dysregulation in cardiac respiratory coupling. We want to use this model to understand better these respiratory mechanisms. For the last 15 to 20 years, we and other groups have find that there are tissue and cell autonomous changes on rabbit models. We can see that we record action potential from atria or ventricular cardiomyocytes. They present prolonged action potential that can lead to long QT syndrome. Also, in very young mice, they present atrial fibrosis. They have altered cardiac neuroanatomy. With all these, we can see they have atrial fibrillation and ventricular arrhythmias. So putting all together, we can say that having mutations on sodium channels that they are expressed in the brain and in the heart make these patients more susceptible to SUDEP. This other area is that we can use animal models is gene therapy. This topic is going to be talked with more depth by Dr. Medford, who is our next speaker. But I want to mention a couple of experiments that they are very important for us. So first, using the Dravet SCN1A animal Dravet model in collaboration with Stop Therapeutics, we inject this mouse on a second day of life, either with vehicle or with the ISO from Stoke, and we saw this very dramatic increase in survival just with a single dose. This first experiment and others done on these mice give the base to put this ISO in clinical trial, who is now in phase 3 and having very, very exciting results. Another model that we are using is using adeno-associated virus to transfect or induce the expression of different genes in mice. In this case, we are using the SCN1B null model that has a very severe SUDEP. As you can see, 100% of them die by the 3rd week of life. But with a single injection of these vectors at day 2, it can increase the survival, and some of them over 160 days that this experiment lasts for. So, and this is also, many other experiments have been done using these treatments. We are hoping with this, we have a good starting or a good idea how to use gene therapies. The final thing that I want to mention, for some of you already seen this morning, is this rabbit model that we are using for studying cardiac, specifically cardiac function. So we can see that because we have been using rat and mouse a lot, but the actual potential for ventricular cardiomyocytes are very different than human. The actual potential for rabbit is a lot closer, and that is because they express similar proteins. And also, they are better modeled because they have similar vitals or closer vitals than human. As we can see here, rabbits are closer than humans than rat and mice. Other advantage is the size. Having a bigger animal, we can have implants, and we can now have 24/7 recordings of EEG, ECG, temperature, and other biometrics, including video, And with that, we can study better what is happening 24/7 in these animals. This model, the heterocycles are very sensitive to heat. And we can see how we can induce seizures with temperature. Only 2 degrees increase in temperature, body temperature, can start an epileptic trace, as we can see here. Also, they can have spontaneous seizures, and those seizures are independent of body temperature. So with this, we can say that animal models are very important to elucidate mechanisms of SUDEP, as we have mentioned, heart rate variability, long QT syndrome, altered respiratory patterns, brain stem dysfunction, etc. Also, it's very important keep using these animals to have pharmacological treatment, gene therapy, and other studies for neuroregulation, hoping these findings can be applied in humans. Thank you for your attention. Also, I want to thank previous lab members and current members of the ISON Lab Laboratory, where we have been working for many years, and also more people, many other laboratories on Michigan medicine and our funding, especially NIH, Dravet Syndrome Foundation, and Stop Therapeutics. Thank you.
Next, we are going to invite Heather Mefford up here, Dr. Mefford, to join us. And it's dark right here. Dr. Mefford is a physician scientist and member of the Center for Pediatric Neurological Disease Research at St. Jude's Children's Research Hospital. Her research is dedicated to gene discovery in pediatric neurological diseases, focusing on epilepsies, with the goal of leveraging information about genetic etiology to develop precision therapies. Her work helped define the genetic landscape of rare developmental and epileptic encephalopathies using cutting-edge genetic technologies. Using engineered and patient-derived cells, her group developed cellular models of genetic epilepsy for characterization of testing potential targeted therapies. She's a science advisor for several patient advocacy groups, co-chairs the ClinGen Neurodevelopmental Disorder Clinical Domain Working Group, and Epilepsy Gene Curation Working Group, was co-PI for the EPI4K Consortium, and is secretary of the board for the American Society of Human Genetics. And I obviously need reading glasses. I will also say I'm one of the, Dr. Mefford is not an advisor to my nonprofit group, but she is someone who is always willing to answer questions, and I consider her a personal friend, and it's my privilege to introduce her.
Thank you for that kind introduction. So I'm here today to talk about opportunities for precision therapies for genetic epilepsies. And I know that not every family in this room was necessarily has a family member affected specifically by a genetic epilepsy, and I'm going to talk about the developmental and epileptic encephalopathies primarily. But I think, I hope that you'll learn from this some of the exciting research that's going on, and I think what we learn from treating the DEEs is going to spill over very quickly into the generalized and focal epilepsies as well. So developmental and epileptic encephalopathies are the most severe end of the spectrum for clinical presentation for epilepsies. And a lot of DEE, in fact, now up to about 50% in the clinic, we can diagnose with a genetic change that's causing the epilepsy. And I think it's important to remember that clinical genetic testing is available for individuals affected by epilepsy, and this is true for all forms of epilepsy. This comes in the form of chromosome arrays, gene panels, exomes, and genomes, which are now the more recommended approaches for genetic diagnosis. And as I just said, the exome testing in the clinic can give us a diagnosis for up to 50% of affected children today. I think genetic diagnosis can be really important for families. It not only gives us an answer and ends the diagnostic odyssey, but it can help us provide better prognosis. So what will my child's life look like over the next 20, 40, 50, 80 years? We can talk about what's the recurrence risk, what's the chance that this will happen again if I have another child. More and more, it affects the choice of medications depending on what the specific diagnosis is. And I think very importantly, it connects families with the same diagnosis who can then come together to discuss their lived experience, to meet with the experts, physicians and scientists, and to actually drive forward research. And for the affected individuals, it often provides research opportunities because, as I'll show you, there's a lot of activity now in the field to develop precision therapies. But to use a precision therapy, you really need to have a precise diagnosis. So I want to emphasize that actually genetics informs treatment today, and this is true for the anti-seizure medications that we have available. And just two quick examples. So we've heard Dravet syndrome or SCN1A related epilepsy mentioned, of course, several times. Dravet syndrome is due to the loss of function of one of our two copies of SCN1A. And so if you can imagine if you've lost the function of a gene that encodes a sodium channel, if you use a sodium channel blocker to further block activity of sodium channels, this may not actually work and may make the disease worse. But conversely, there are other sodium channel genes where the mutations actually cause gain of function or increased function of those sodium channels. And in that case, the use of sodium channel blockers actually works, and you want to use them. So understanding the genetic diagnosis for two very different conditions that may look very similar when the first seizure happens, is important to get the treatment right. But these are treatments. They're aimed at reducing seizures, which is extremely important, but they're not cures. And we think that with more and more information about genetic diagnoses, and importantly, better tools that we have today, which I'm going to tell you about, that we can do better. So there are many new genetic technologies on the horizon, or not even on the horizon, but here. And I'm going to talk in a little bit in depth about a couple of these and just mention a few at the end. But these include gene replacement, or what we kind of think of traditionally as gene therapy, antisense oligonucleotides, which Dr. Luis Santiago just mentioned, and I'll touch on as well. CRISPR approaches. Everybody has heard about CRISPR, so one of the first questions I often get when I give a diagnosis is, can we use CRISPR? Probably not, but I'll tell you about how we're thinking about that. And then several others like cell transplantation and small molecule therapies and drug repurposing. But I want to start with a little bit of basic genetics. I promise it won't be too much, because understanding the genetics helps us figure out which therapy might work best. So many genetic epilepsies are due to a genetic change or mutation or variant that happens on one of the two copies of our genes. We all have two copies of every gene, one from mom, one from dad. So a mutation in one copy of that gene causes it not to work. So you have one working copy and one non-working copy. You can also, these often occur de novo actually, so this is often how something like Dravet syndrome or CDKL5 might happen. We also have recessive conditions where you have a variant that you inherit from mom and a variant that you inherit from dad, which causes neither copy of the gene or the protein to work. Or you can have a mutation that occurs similar to the first example I gave, but instead of causing the gene or the protein not to work, it actually causes it to work differently. And the way that it works can either be a gain of function, like I mentioned for those sodium channels, or perhaps it has a new function that gets in the way of the activity of the normal copy. So just giving back the normal copy may not actually fix the problem. So for each of these, we have to think about different approaches. For the haploinsufficient or the first example, we want to think about either making more of that wild type or normal copy, or just replacing that broken copy. If we have a recessive condition where neither copy works, you got to add back some a normal copy of that gene or protein. And in the case where it works too well or is causing problems because of the mutation, you actually need to get rid of that copy, which we think of as toxic to the cell. So very different approaches depending on what the actual condition is. These gene targeting therapies that I'm going to talk about are different from what we have available today. Although we have great anti-seizure medications, physical therapy, behavioral therapy, medications for behavior, these all are aimed at one or more features of the disease, but none of them treat all features of the disease. And we think that perhaps using gene targeting or gene modifying therapies will help us get at the root cause and perhaps treat the condition in whole. So the first one I'm going to talk about briefly is gene replacement or gene therapy. This is an approach where you actually add back a working copy of the gene where the mutation is causing epilepsy. This is the SCN1B example that Dr. Luis Santiago just mentioned. To do gene therapy or deliver a working copy of the gene, you need to package it to get it into the cells. And to do this, we package it into a viral particle, and usually it's adeno-associated virus, or AAV. And these viruses actually, when we're thinking about epilepsy, they're actually able to reach the brain, which is great, and deliver the gene. And there are actually quite a number of gene therapies that are FDA approved in clinical use today, not for epilepsy, but for spinal muscular atrophy, for example, which is a neurologic condition, for Duchenne muscular dystrophy, and then for several other non-neurologic conditions as well. This approach we can think about using for those haploinsufficient or dominant disorders, or for recessive conditions where you really need to put back a working copy of the gene and protein. The advantages of gene therapy is that really it's a one and done therapy, so it's a one-time treatment. It's generally safe, though there have been, certainly have been complications and adverse effects that are important to know about before you embark on this treatment. There are many challenges to gene therapy. If there weren't, we'd have lots of gene therapies available today for many of these conditions. We have hundreds of genetic epilepsies, and I wish we had a gene therapy for all of them. But some of the challenges are that you have to package that gene into a virus particle, like I said, and there's a certain size limit. So some of the epilepsy genes are actually too big to package this way. We also need to think about delivery. So how do we get that working copy of the gene to the brain and to the right cells in the brain, and how do we know what the right cells are and how many of those cells we need to target? And then the other important consideration is that it's a one-time therapy, but it cannot be reversed. And so if there are side effects, there are ways to treat those side effects, but there's no way to take that therapy away. Unlike an anti-seizure medication that you can stop if there are side effects, you can't do that for gene therapy. So there are several different ways to think about using CRISPR as an approach for genetic therapy. The first one is kind of how it was first thought to be used, which is gene editing. So can we use CRISPR? CRISPR is an approach that actually allows you to go to a specific place in the genome. So you can think about targeting the specific mutation that an individual has, and you can go in and tell the cell to fix that, to change that genetic mutation. This is the way it was first used. It actually goes in and cuts the DNA and then repairs it in a way that fixes the mutation. There is one approved CRISPR therapy today on the market, and this is for sickle cell anemia. And in this case, what you do is you take out the stem cells for blood, actually treat those in a way that you fix the mutation, or there are some different approaches, but it uses CRISPR to fix that sickle cell mutation. And then you can put those cells back into the patient. You give them back to the patient. You take out their own cells, you fix them, and you put them back as stem cells, and then they produce healthy blood cells, curing sickle cell disease. So this is really exciting, and it's, I think, going to change the landscape for sickle cell patients with sickle cell disease. One of the challenges for epilepsy is it's a lot harder to take out stem cells for neurons, fix them, and put them back. In fact, it's not possible right now. I think we can think about ways to get creative about thinking about ways to do this. But the other challenge is that you could think about, okay, we can't take cells out and fix them and put them back, but can we deliver that CRISPR machinery to the brain and say, go to those cells and fix the mutation, and the cell may work normally. So this is something that people have certainly thought about and are working on. One of the challenges is, again, that you would have to deliver the CRISPR machinery as well as tell it where to target, and that's kind of bulky machinery. Just like some genes are too big for gene therapy, this is a similar delivery approach. So people are working actively to think about how can we do this efficiently with new technologies. The other ways you can use CRISPR is not to go in and cut DNA and fix it, but is actually to go in and send it in with an attachment that activates gene expression or turns a gene on. And so in this case, it would be CRISPR-A, A is for activation, to increase the amount of protein that's made. And again, we can think about using this approach where there's not enough of the normal protein. So if you have a mutation on one copy, but you have a healthy copy that's still there, and you can go in and tell it to make more, this may be an effective approach. This might be also an effective approach for some recessive condition where the gene still works a little bit, just not enough to be healthy. And then conversely, you can put a different attachment on the CRISPR molecule that actually turns off gene expression. And this would be important for those disorders that have a gain of function or a toxic a mutation that causes a toxic protein for the cell. And if you can go in and turn off the production of that toxic protein, it may be an effective therapy. And again, some of the challenges for treating epilepsy with these approaches would be that we need to deliver the CRISPR machinery to the brain and to the right cells in the brain. The other thing to think about, which is a nuance perhaps, but you can't, this approach does not actually target a specific mutation, so not the mutation copy and not the normal copy. It works for both copies. So if we go in and turn off this gene that's making a toxic protein, it's actually going to turn off the toxic mutated copy, and it's going to turn off the wild type copy, and that may actually cause a different disease. So we need to get creative about how we use this as well. Perhaps a more promising approach today is antisense oligonucleotides, or ASOs, which you briefly heard about. So ASOs are little pieces of RNA that you can tell it exactly where to go in the genome. So you can target specific genes or mutations in the genome. And depending on the bells and whistles that you add to these ASOs, you can use them to change how genes are assembled, or the splicing machinery to actually skip a mutation, for example, and still make a healthy protein. You can also think about using it to actually, again, to increase the production of a healthy protein or to turn off the production of a toxic protein. And I'll give you a couple of examples. So some of the advantages of ASOs are, one, that they're easier to deliver, so I didn't put that on here, but it's true. There are also several ASO therapies that have been FDA approved and are in use today, and this includes one for spinal muscular atrophy, for example. And importantly, this is a therapy that you give that is not permanent, so if there are side effects, you can consider stopping treatment. One of the challenges is that it's not a one-time therapy. So the ones that are in use today for neurologic disorders are given about every 1 month to every 3 to 4 months, depending on the ASO. So it has to be delivered repeatedly. We don't know for how long. And often for neurologic diseases, this is done by lumbar puncture or a spinal tap. So you already were introduced to ASO therapy for Dravet syndrome, but I'll give you a little more information on this. So again, Dravet syndrome is caused by a genetic change in one copy of the SCN1A gene, causes it not to work. You still have a healthy copy. So the goal here is to use the ASO to ask, can we increase the amount of that healthy copy that's made and provide enough SCN1A to the cell to be a healthy working neuron. So the ASO treatment that is being used today actually changes splicing, or how that SCN1A protein is made. And the end result, I won't get into the nuances, but we can talk about it after if you'd like, is that it makes more of that normal, healthy, wild-type SCN1A protein. This ASO was, all the preclinical work, as you heard, was done by Dr. Isom's lab, showing that it's very effective in a mouse model of SCN1A. Stoke Therapeutics has done a phase 1/2A clinical trial of ASO for Dravet syndrome. The drug is the ASO is now called Zrevanersen, I think. And it's delivered by lumbar puncture every 3 months. Their initial clinical trial data is very encouraging, shows that there's a significant reduction in seizures in children who are treated, and that there are improvements in cognition and behavior for those aspects that they have been able to measure. This was known as the MONARCH study. And now I think I've just heard that they've been approved for a phase 3 trial as well. And there's a lot of, for those of you who might be at AES, I think there will be several presentations or posters about those results. There's also an ASO therapy that's in clinical trial right now for SCN2A related epilepsy. So the difference here is that the genetic change in SCN2A that causes developmental and epileptic encephalopathy is one of those that leads to gain of function of the SCN2A protein that carries the genetic change. And so we need to get rid of that. We can't just add back normal SCN2A. And so in this case, the bells and whistles that you add to the ASO cause it to bind to the mutant copy of SCN2A, and then it recruits other factors that chew up that RNA and say, do not make protein. And so you're getting rid of that toxic copy of SCN2A, and maybe a little bit of the normal SCN2A, but that seems to be okay, and to treat the disease. They're not quite as far along as far as results, but there is a clinical trial that's being run by Praxis, and they have treated several SCN2A patients. This has been delivered by lumbar puncture about 4 weeks in this case for safety data. The safety data that's been reported so far is very encouraging, and the preclinical data suggests that it will be effective for seizures, but these trials are ongoing, and I think there will be more data coming out soon. And if anybody out there has additional information, I'm happy to hear it. So those are two examples of ASO therapies that are in trials, and I think there are many, many more in research laboratories right now being tested. So our lab is actually working on 2 or 3 different genetic epilepsies using ASO therapy, as well as 1 or 2 genetic epilepsies using that let's turn on and make more of the gene type therapies. And there are many labs doing the same thing for different genetic causes. I just want to mention that there are several other emerging therapies, that some of which are more specific to a genetic disease, some of which are maybe not for a specific genetic disease, but for epilepsy. The first being cell transplantation. And what I mean by this is taking healthy, usually inhibitory type cells, so cells that kind of restore that excitatory inhibitory balance to treat seizures in the brain. This has been shown to be effective in mice. There's actually a sea lion that's been treated in San Francisco by this method that is now seizure-free. And there is I believe there's a clinical trial, and someone can correct me if I'm wrong, but using this approach to put healthy stem cells that they hope will develop into inhibitory cells into brains of some individuals who have focal epilepsy, and this is in humans. So this is being considered as a potential treatment for epilepsy. It's not specific to any gene, just specific to epilepsy. Small molecule therapies are another approach where this is not, is gene or pathway specific, but these are small molecules or drugs that are being developed that are designed to interact with specific genes or pathways that are affected in either genetic epilepsies or pathways that we know go awry when seizures occur. And then finally, drug repurposing is an approach that many individuals are working on in the lab, and this is screening drugs that have already been FDA approved for something. It doesn't have to be epilepsy, and using either a cell model or a animal model to test a large number of drugs in those models where there's a genetic change causing epilepsy, and asking which ones correct the problem or prevent seizures from happening. And so sometimes we're surprised by the things that come out of these. And one example is fenfluramine for Dravet syndrome. Fenfluramine was one of the drugs in fen-phen years ago used for weight loss, but is now approved for Dravet syndrome and seems to be working for some individuals. So I've given you a few examples of, I think, what are exciting technologies that are ready for epilepsy. But I think there are important considerations to think about, because we're all very excited about this, but we have to remember that we need to benefit patients and do no harm. We also need to think about the best delivery system and how do we make this the most effective for patients. So some considerations that I think are worth thinking about is, again, how do we deliver the therapy, no matter what the therapy is, to the brain? There's a blood-brain barrier. It's there for a reason, to protect the brain, so it's hard to get things into the brain. So right now, this is often done by lumbar puncture, but we need to think about other delivery systems that might circumvent that. And also, how do we know what are the right cells in the brain to get to and how many we need to treat to have the best effect? We think a lot about the brain because epilepsy is a brain disorder, but there are syndromes that that cause more than just epilepsy and have other effects. And I think when we think about preventing SUDEP, we need to think about, are there other tissues that we need to target? We've heard a little bit about the heart, and some of these genes where mutations cause epilepsy are expressed in the heart as well. So do we need to be thinking about delivering it not just to the brain, but to other tissues? And this might be true for other syndromes where they have heart defects or hypotonia. Do we need to get it to the muscles, this type of thing? Again, as I've already mentioned, some therapies are permanent, so we need to think about what are the side effects, possible side effects, how do we treat those, and will we be able to either prevent those or take therapy away if they occur to prevent unwanted harm to patients. So I'm going to stop there. I'm happy to take questions during the panel session, but I hope I've convinced you that genetic diagnosis is available, it's important, it's informative, and that's today, even when there's not a precision therapy available. Because it can inform treatment. And then I think these tools that are coming down the road, and there are many more that I didn't talk about, but are similar ideas to turn genes on or off or fix mutations that I think offer hope for patients living with epilepsy. And I think this is true, especially for the DEEs right now, but this is very quickly, we know that some focal epilepsies are caused by genetic mutations, some generalized epilepsies, and I think quickly this is going to spill out into the broader field of epilepsy. So thank you for your time, and I'll take questions when we're done.
Thank you, Dr. Mefford. Our next speaker
Hello,
is...
thank you so much.
Me? Our next speaker is Dr. Sandy Lamb, who is Chief of Pediatric Neurosurgery at Lurie Children's Hospital in Chicago, and she's joining us through a recorded talk. Let's see.
Hello, thank you so much for allowing me to join today and share some thoughts about surgical treatment options, which can be tools to reduce the risk of mortality in epilepsy. So we know that epilepsy affects so many people in the US population, and this has been the theme of the conference, that it really can affect anybody. I'm a pediatric neurosurgeon, so I'm focusing more on the children affected by epilepsy. And here we're looking at drug-resistant epilepsy when you may be eligible to see a surgeon. So usually we would look at the National Association of Epilepsy Centers for multidisciplinary care. And when you look at anti-seizure medications and the chances of seizure freedom with each successive anti-seizure medications tried, we know that the chances of obtaining seizure freedom with each anti-seizure medication gets less and less. We also know that living with epilepsy is dangerous, and that especially in children, the mortality rates of living with epilepsy are higher than children without epilepsy. So from a neurosurgeon's perspective, the rationale for epilepsy surgery is that there's a role to be evaluated for surgery because knowledge is power, and there's a role for surgery when it can be performed safely and effectively, because ongoing seizures have detrimental effects on the developing brain, and that's from the seizures, that's from the medication side effects, and that's from the risk of death in epilepsy. There's also developmental stagnation or the regression of milestones from kids living with seizures, and also that children have plasticity of the nervous system, so they're able to remodel and develop their brains in ways after epilepsy surgery. So when you look at surgery, when you look at consensus recommendations from an expert panel, referral for surgical evaluation, not surgery, but just evaluation for epilepsy surgery should be offered to every patient with epilepsy across a spectrum of ages when patients have drug-resistant epilepsy. So even so, when you are having seizures, even on medications, every family should be empowered with the knowledge that this may be an option, because poorly controlled epilepsy is dangerous. So when we talk about surgery, there are so many surgeries that are discussed with different terminologies and different options. And there's really an evolving role of the things that we can do in surgery now. But keeping in mind that when we look at all of these options, some patient cases will be relatively straightforward and some will be very complex. But everybody deserves a chance at being evaluated when you have drug-resistant epilepsy, because when you look at the most ideally selected patients, a majority undergoing epilepsy surgery can have seizure freedom. And of course, we continue to try to do better with better surgeries, better research, and really try to offer better quality of life and better outcomes. So when we look at surgery, there's actually a lot of innovations and a lot of advances that have happened even within the past 2 decades. So people who had been considered not candidates for surgery before actually can be candidates now, because there's so many different options, different technologies, and a lot are more minimally invasive. And we also have the option of neuromodulation now, where there are different devices that can help with different types of neurostimulation and neuromodulation. So when we look at how long we've been doing this, it's been over 2 decades, where there's a randomized controlled trial for temporal lobe epilepsy showing that surgery compared to continued medications actually has a significantly higher chance of seizure control and seizure freedom. This was published in the New England Journal of Medicine. And there's been decades of research showing that we can do this in adults and we can do this safely in children. And here's an international consortium showing that even in infants up to 3 months of age, we're able to show that we have good seizure outcomes and good safety profile. I'll share some of my own research, which uses a national databases of different sources. Here I'll show you something from the Children's Hospital Association. This is an older study, retrospective, looking at over 45 pediatric hospitals data. And we looked at patients with drug-resistant epilepsy and looked at those who continued to get medications and added a medication or got surgery of any type. And when you look at different outcomes, such as the medications they're on, going to the emergency room, being admitted to the hospital, or survival at 2 and 5 years, there are significant differences. So when you look at this in a different way, we can look at utilization, which is coming to the hospital in the emergency room or in inpatient visits, coming to any care, inpatient or outpatient, for seizures or otherwise, and then being on anti-seizure medications. The surgical groups did better in terms of not needing to have as much contact with the healthcare system. And there's this hint that we saw in this older study that there's survival rates were better in the surgical group, but this was only at 2 and 5 year follow-up. So I'll show you some extensions of that. coming up. But when we talk about outcomes in epilepsy, this whole conference is really talking about mortality in epilepsy, and how do we think about that, and how do we think about that risk. And then when you think about other outcomes reported are seizure reduction, healthcare utilization, how many medications people are on, neurocognitive development, especially in children, quality of life, and survival. So when we look at this in a longer follow-up using the Children's Hospital Association database, we developed a cohort where we had over 18,000 patients selected from this database with over 10 years of follow-up. So we looked at the probability of being alive at 10 years, looking at pediatric patients living with drug-resistant epilepsy in this database. And we looked at those who had continued medical therapy, vagus nerve stimulation, or any cranial epilepsy surgery. And when you look at the survival curve here, you can look at the difference between the medically treated cohort, surgically treated cohort, and the vagus nerve stimulation cohort. And those with the medical treated cohort actually had the lowest survival at 10 years. So if you look at this here, have this shown a little bit bigger, that there's a statistically different, significant difference in the survival curves of the cohorts who are treated with brain surgery, with VNS, or with continued medications. So in numbers, I'll show that the unconditional probabilities of survival beyond 10 years for the anti-seizure medication cohort is 89% or almost 90%. Those with anti-seizure medications and vagus nerve stimulation is 93%. And then those who had brain surgery or cranial epilepsy surgery while on medications, had a chance of being alive at 10 years of 98%. So when you look at these numbers, the difference in the chance of survival at 10 years is very different between the medication cohort, vagus nerve stimulation cohort, and the cranial epilepsy surgery cohort. If you look at it a different way, with the same numbers and the same cohorts, so compared to being on medications only when you're on when you have drug-resistant epilepsy, the risk of overall death was reduced by 35% in those who had vagus nerve stimulation, and reduced by 83% in those with cranial epilepsy surgery compared to those on medications only. So, this was the commentary and editorial in our publication in Lancet Child and Adolescent Health, and this is from the Toronto group, from the epileptologist and the neurosurgeon, thinking, well, can we think about this in a way where we think about drug-resistant epilepsy and the chance or the choice that we have to continue medications or to think about surgery? Can we reduce the risk of mortality in well-selected patients, or in the way that we approach how do we offer surgery versus medications? And is there a survival advantage in the surgical cohorts? So when you think about this from a large kind of numbers and public health perspective, the opportunity for preventable deaths in epilepsy may be actually larger than the number of preventable deaths from the big public health campaigns such as sudden infant death syndrome and sleeping on your back and putting babies on their back, or fire prevention for stop, drop, and roll, and using the fire extinguishers and reacting appropriately in fires. So how do we think about this more? When I've shown studies that show that epilepsy surgery is effective, we also know that epilepsy surgery is underutilized. So, I'm a surgeon giving a talk, but, I think knowledge is power, which is why I'm giving a talk in this way, saying that the opportunity to be evaluated for surgery is power. that patients, children, pediatric patients with drug-resistant epilepsy, in our study who underwent cranial epilepsy surgery or brain surgery or vagus nerve stimulation had higher survival rates than those who received medications only in this big cohort of over 18,000 patients. So, is there a role for increased access to surgical evaluation and surgical care so that families have that understanding and can have individualized attention and evaluation to see if they may be candidates for surgical treatments or other types of treatments? Because we know that an evaluation at a comprehensive epilepsy center and having a team-based approach to see what could be effective at helping seizures and reducing the seizure burden. Whether or not somebody gets surgery, actually, that evaluation can produce better outcomes. And then I've shown that actually surgery can also produce better outcomes in very powerful ways. So I'll leave you with a few thoughts of, even though I've shown that epilepsy surgery is effective, we know that it's underutilized. In America, we know that about 1 in 3 patients who may benefit from epilepsy surgery, who are children, actually get surgery. And many actually don't get surgery. And that's a majority of kids living with drug-resistant epilepsy who may be candidates to benefit from surgery. We know that this isn't actually only in the United States. This has been reported in different timeframes and in other countries. And when people ask why, actually my own colleagues asked why, and in my previous practice, we said, well, let's look at our own practice. We looked at almost 100 patients with the same diagnosis of cortical dysplasia, and we looked at who got surgery. And then we looked at what were the factors in the journey that led them to surgery. And we found that the time between the diagnosis of drug-resistant epilepsy to actually getting surgery for the same diagnosis of cortical dysplasia was very, very different. And patients of non-white race actually lived for a longer duration with epilepsy before getting epilepsy surgery. And that difference was over 3 years. And these patients were more likely on Medicaid and actually lived closer to the hospital. So when we look at our own hospitals and our own practices, where are the barriers in care that families can face or that healthcare systems may have? And we can look at, referrals. We can look at access to care. Are there socioeconomic factors, cultural factors, health literacy, language, and really a lot of different things that can go into access to care. So here, when we look at what's ideal when you have epilepsy and get the diagnosis and the referrals and being on medications and thinking about understanding if you're a candidate for surgery, we know that reality actually is that very few patients are seen at multidisciplinary care centers and the comprehensive epilepsy centers. And we also know that while many people can be candidates for surgery, very few people are actually getting surgery. And when we look at the delays, when we look into the adult population, we can see that this journey can take 1 to 3 years in a short time frame for children, but almost 20 years for certain adult populations in the literature. So, and then of course we follow patients after the treatments. But when you look at, different time points in people's epilepsy journeys, we can actually see that from diagnosis to referrals, there are multiple ways that people report that there have been delays noted in how we actually help patients and families get through the medical system. So there are many other causes that we don't know quite about yet, that we haven't quite characterized, that can be socio-demographic causes or barriers in access to care. And we need to understand that a lot more. And that's why in my research, that is when I'm not doing surgery, I've shown you different types of ways that we try to use big data. And now in my current study, I'm actually working with a multidisciplinary team and with the Lennox-Gastaut Syndrome Foundation to look at patient-centered outcomes. And we're looking across multiple centers. And here we're looking at comparative effectiveness of palliative surgery, such as corpus callosotomy or VNS versus additional anti-seizure medications for LGS. And this is because families have told us that this is something that they face. You know, do we add medications? Do we keep trying this, or should we really think about surgery? And when do we do that? So this is the first nationwide study that we're looking at this question that families pose to us, which is, this is a common question that we face, and we want to have good data for this. So this is our core team, a pediatrician and medical informaticist, Mark Rosenman, Anu Patel, pediatric epileptologist, and Tracy Dixon Salazar of the LGS Foundation, who's also an LGS mom and a neuroscientist. I told you we're looking at comparative effectiveness, which is the way that we look at things that we know how to measure, which is, do you go to the ER? Are you admitted to the hospital? And families saying that we want to measure things that matter to our daily lives, like communication, behavior, and quality of life. After we have this choice of medication versus surgery. And then, of course, we'll look across the country at our national data to look at the real world utilization of actually who gets what therapies. And then we look at where the variations are and where the disparities are and where we can do better altogether. And when we've worked with families to understand what they want to know and what matters, is that we're seeing a lot of gaps that as surgeons and as epileptologists and as doctors in the healthcare system, we don't take the time, or we're not good at measuring what families want to know about communication and if surgery impacts that, or behavior, or quality of life. And here, you know, we need to talk about mortality and survival more, and how do we measure that, and how do we talk about that risk? And I'm excited to report that our next steps and our next grant with our PCORI sponsors is actually to measure indirect costs of care and caregiver burden to families. And what does it cost to families to take care of children with epilepsy? What do they give up? What do they wish they knew and what they had planned for? And this is economic to families, but this is also along the care journey of the patient experience. And in doing so, you know, it's not that families can't make it to appointments or that they're called no-shows in the healthcare system. It's that there are siblings, there are jobs, there are elders in the family. And how do we balance all of that? And then how do we as healthcare providers, surgeons, epileptologists, researchers, understand how we use this information together to redesign the system for better care and support, and also to understand the risk of mortality, and how do we be able to talk about that all together to empower families. So this is something I'm very grateful for your time, that I can talk about this as a surgeon and a researcher for how do we think about the role of surgery as we think about the risk of mortality, and really how do we use surgery as a tool to think about treating epilepsy and thinking about being partners against mortality and epilepsy together. Thank you so much.
Our thanks to Dr. Lamb for her excellent talk. Our final speaker is Dr. Nathan Fountain, who is the director of the Comprehensive Epilepsy Program at the University of Virginia. His research focuses on human experimental therapeutics, particularly drug and device treatments for epilepsy, and he is a founding co-chair of the Epilepsy Foundation's Research Roundtable for Epilepsy. So welcome to Dr. Fountain.
Thank you, Tom, and everyone else for inviting me to speak, and thank all of you for staying. I think I have the honor of being the last speaker. so I'll try to get us through to the end on time. And I especially want to thank all of the families who have spoken here. Every time I hear a description of anything like this, Jenny Symes spoke at the Research Roundtable for Epilepsy that one of the co-chairs, Tom, said, it gives me the same kind of sinking feeling in the pit of my stomach. My heart races, and I think of all those conversations I've had with families, unfortunately, which I've had 38. So we have a database of patients with epilepsy that have 3,300 or more patients with epilepsy, and 38 people have died of SUDEP. So it's something kind of makes me anxious because it's such a traumatic thing to hear about and even more traumatic to experience, of course. So my charge today is innovation, access, and devices, and where are we today? But really, what I'm going to talk about is the findings from the Research Roundtable for Epilepsy. I'll talk briefly about innovations, and the reason I'm only talk briefly is because we heard quite a bit about innovations, and then spend most of the time on access, which is really important because we can have the best ideas in the world, but if they can't find their ways to people living with epilepsy, they're not going to do anybody any good. So my day jobs, I'm an epileptologist. I take care of people and families with epilepsy and do clinical research. And the two places that I see the system, as probably most of us here, is sort of at the beginning of the system, research and innovation ideas, and at the end of the system where we can provide those innovations or devices, as I'll call them, or technology, to people with epilepsy. But in between is a huge gap. For drugs, the thing we most often think about, the gap is between when someone comes up with an idea and then needs funding or opportunities to get it to people. And once it's first in people, it goes off to the races. And of course, we think about drugs in all aspects of medicine, including epilepsy, every day. It's an established pathway. But devices and technology is brand new in many ways. It has a different kind of gap, partly because it's relatively inexpensive to come up with innovation and to bring it almost to people. And so I'll talk about some of the problems with access that are maybe not what you'd considered. The innovations we already talked about that have important drivers that are unique and new, why devices are different than drugs. You know, the fact is vitally needed is just self-evident from the fact we're having this meeting, of course, but we have new technology, new ways of thinking about things. And that's illustrated over here on the side of the screen in this diagram about brain-computer interface, BCI systems. And from this article by Crank, you can see that some of the things that are so different or new, at least in my time frame, is to think about dry electrodes, which means just a different way of recording EEG for a prolonged time. About EEG amplifiers, they call it. I'd probably call it different kinds of hardware. And then firmware, the ways these things interact. And across all devices, this is true, but in epilepsy, we have more opportunities than ever before. And also, we have some payer acceptance of that technology. And so payer just means insurance. There's different kinds of insurance, but I'll call them payers because it isn't necessarily insurance. And there are some things that Medicare and private payers and others will pay for. And so that could be a driver of innovation, just like it is for drugs, for example. The first kind of hurdle I want to talk about is regulatory hurdles with the FDA. And this may be where you would start. So in drugs, we often think about hurdles through the FDA. And in fact, the Research Roundtable was founded to overcome some of those hurdles. And it is true that there are some hurdles in device development and a different way to think about it. And so I have to set a little background information about what those hurdles might be. And there are two basic regulatory pathways for devices to be approved by the FDA. One is a de novo device or pathway, meaning something that someone thought up that hasn't been done before, or a predicate device pathway in which a device exists. And so someone's already found an indication for this device, it's already got it approved, that means it has demonstrated safety and effectiveness, and now the FDA and everyone else will use that pathway based on that predicate device to approve that device. When there's a lack, the problem with the de novo device, which is what we want, right? We all want some brand new thing that can both detect seizures, predict seizures, and intervention to prevent seizures, and prevent other epileptic activity that prevents SUDEP and makes people better. So that would be a entirely novel device. We don't have that yet. That's what we're after. But at every level of that, really there's lack of established scientific basis for all those devices, or almost all, except the few I'm going to talk about. A lack of established measurable clinical outcomes. And this might seem obvious. The outcome should be SUDEP or death. The problem is, although it's not a rare event, it's rare enough that it makes clinical trials very difficult to do, or difficult for the FDA or regulators or any of us really to understand how you might test that in a realistic time frame. And finally, we have a lack of established measurement tools for these outcomes. Whereas for a predicate device, something that already exists, think of an EEG machine, all you have to do is demonstrate that it's just like the one that already exists or is better. So that's a much easier pathway. And those are all hurdles you could overcome. But the hurdles in business development, as I'll call them, are much greater. So obviously, funds are necessary to develop devices, and that means they have to attract investors. And for most investors, for instance, think about drug development for a minute, because that's kind of what this is perpetually compared to. There are methods of risk mitigation, ways you can go to an investor and say, well, here's why we think this is going to work and why your $10 million is going to be useful in helping us find the answer. And usually you can establish an overwhelming clinical need, and that's pretty easy in our case. And also follow an established regulatory pathway, so you can tell investors and everyone else, look, there's a pathway ahead through the FDA that's been done before. We're going to follow that pathway. And you can establish a reimbursement method. So you say, we're making drug X, for instance. It's been used for seizures. We know there's a way that we can ultimately get that to people and can have a business model that can be successful, for example. And there's different methods of payers that we'll talk about. So finally, you have to have a plan that's profitable. If you ask somebody for $10 million or $1 million or $20, they're still going to want to know that in the end there's some kind of payoff for that. So that means you have to have an established plan for not just conception, but through FDA approval, through insurance reimbursement, and to sustain marketing, a commercialization strategy, or whatever you're going to do to finally have the device available to people with epilepsy. So overcoming these hurdles is a challenge. That's why they're hurdles, of course. And about 10 years ago, we established the Epilepsy Foundation's Research Roundtable for Epilepsy, or REE, which has been very effective in looking at hurdles for drug development. So over the past 10 years, almost each of the RREs has led to a change in FDA policy. So for example, there was a time when you had to study epilepsy drugs in adults and do it all over again in children, and that's very difficult to do in children. So partly as a result of the RRE, the FDA now allows extrapolation for those drugs. You don't have to repeat all that in children. Or they now allow a different clinical method for studying seizures in clinical trials called a time to nth seizure that reduces the time that people are in the placebo arm, which is really important because that's when SUDEP occurs and death of other causes. So last year we convened the first diagnostic and treatment RRE, which really you can think of as a device RRE, which is really what it's turned into. And that brings together regulators, so the FDA, the EMA, that's the European Medicines Agency, drug developers, or in our case, device developers, and topic experts, and tries to identify a single topic or limited topics to overcome. In April of 2024 was the first device RRE. And of all the hurdles, that means hurdles in innovation, hurdles in science, hurdles in FDA approval, hurdles in commercialization and payment, of the 27 member companies who came, they almost overwhelmingly identified that the hurdle was commercialization strategies, or what I would call reimbursement. So that was more concerning than anything else, including the science, more difficult than all the other aspects of developing their devices. And so while this wasn't something we'd ever conceived, ever thought of, we were really occupied with the science, it turned out that we spent a lot of time talking about reimbursement methods or payments, because that's really the hurdle that most of them have found. And the reason this is so important is because the payment method that ultimately is expressed depends on the type of approval. So that for, I guess if we skip ahead to the different considerations of types of approval, for the predicate pathway, so that's when a mousetrap already exists and you're just building a better mousetrap, to sort of speak. That makes sense because it's the fastest pathway with known FDA policies and known reimbursement methods. But the problem is it follows the established reimbursement method, and for devices, the established reimbursement method, such as it is very low. It also means the devices aren't that novel, so that's a problem for us in taking care of patients and people. The de novo pathway, on the other hand, sets its own performance characteristics. So in other words, you can certainly define it as better. You can look at something different that hasn't been looked at. Everything can be novel. and there isn't already a low reimbursement level set. On the other hand, it might be difficult to achieve those performance characteristics. You might have set yourself a pretty high bar. We hope that that's achieved, of course. And it's unknown whether it'll be reimbursable. So for reasons I'll talk about in a moment, that can be very complicated. And then finally, you could decide you're not even going to pursue FDA approval. Your device is going to go outside of that. There are devices that treat health or detect health, like fitness trackers, things like that. If it doesn't diagnose or treat disease, it could be treated as a health app and go outside of this and just go by direct to consumer. That has sort of obvious advantages, and then the disadvantage, it may not be so obvious, it has an unknown patient uptake. So for reasons that are mystery to us all that we heard about earlier today, there's not as high a patient and provider uptake for the existing devices that we have now. So that's a potential hurdle. So if you think about the pathway from conception through actually getting into people's hands who can use a device, you might think about it in these steps. And if we think first about the sort of standard predicate approval, the way that a mousetrap already exists, so a device for detecting seizures, let's say you're going to follow that through a predicate pathway. Well, the first thing that happens after FDA approval is it's assigned a CPT code, which is a specific code that's used for billing. And you use the existing CPT code, and that's easy, because you say, we already know it for this, we're going to use it for that, for this new thing. And then CMS, the Centers for Medicaid and Medicare Services, so Medicare says, okay, we know what that device is, we know about it, we have this other method, these other codes I've listed here, we can know how to pay for that thing. And then Medicaid and other payers follow Medicare typically. Private insurance comes along and says, okay, I see how that works, we'll just do the same thing. And the business model for that really depends on the device payment. And what it means is that you're going to sell enough of your widgets, enough of the devices to make the whole business work in the end at the payment level that's already been specified by those CPT codes, which are really specified by Medicare. Whereas for the de novo pathway, the one in which you've invented a new device, you're going to strike your own pathway to approval, then you have to create a new CPT code. Creating a new CPT code is an arduous, complicated, long experience. And many of the things I'm talking about right now are very arcane, not really have anything to do with patient care, things that we don't ever think about as clinicians, but are kind of vitally important for getting from the beginning to the end. And this is a step that's particularly long and arduous. And when it does happen, in the infinite wisdom of Medicare, typically a particular kind of CPT code is assigned to this device called a CPT-3 that's often not reimbursed for 5 years while it's being researched. That is, it's being given to people to show it's actually beneficial, or is reimbursed at a certain level below what they think the cost is. And the problem with that's kind of obvious. You're just giving the device away, no way to sell it for the first 5 years. And there are people who have done that and made that work. But more often, you'd like to think there's some other way to keep your business alive for those 5 years and continue to get the device available to people with epilepsy, for instance. So this is just an example of the complicated schemes you have to go through for any of these new assigned codes. The details don't matter, except that almost all these circles end in denial with a few approvals, which is sort of the bureaucratic way. So I just want to mention two CPT codes that do exist. These are the billing codes. The details don't really matter, but the idea is if we were to look for something we could use to find a path forward, these are two. One called remote physiologic monitoring, the other called remote therapeutic monitoring, and they have their own unique qualifications. You might think the name remote therapeutic monitoring is perfect for us, right? Except it only applies to respiratory and musculoskeletal system for other obscure things, so not neurology. The remote physiologic monitoring, the RPM, which also might make sense, also has certain requirements that are difficult for what we're talking about. So that means they don't work well for the kind of devices we've talked about, such as daily monitoring to detect seizures. And then in the end, you'll see the reimbursement's $48. So that's quite a complicated process. And I just wanted to mention briefly some ongoing device challenges that have actually been overcome. So many of you are aware of what we've talked about several times or alluded to, a wrist-worn seizure detection watch from Empatica, which one of the challenges is really a lower uptake by providers and patients than expected, at least than I'd expect, and I think everyone would expect. Insurance denial. and then one win, so one counteraction to this, and that's the Minnesota Medicaid has a policy, really a model policy, for local coverage of the device. And this is a fundamental shift in how to think about things. So it just skips Medicare, goes right to the local state Medicaid level. And that is a path forward that has a lot of opportunity to it. The other is a simple limited EEG electrode application for status epilepticus called Cerebell. I think we heard a little bit about that along the course of things today. And here the strategy is the whole device is purchased by the hospital for emergency use, takes it out of that whole other system I was talking about. Now we don't have to worry about all that. Just like the hospital purchases OR equipment but doesn't charge anybody for it, it's just part of the whole process of care. And there's a new technology add-on payment available for 5 years in that particular setting, and that's allowed that system to move ahead. And then finally, there's an eye movement detection device from eyes that actually doesn't diagnose or treat illness. Instead, it just detects eye movements, but it so happens that seizures have a lot of eye movements with them, and so it could be used for that, among other things. And that's a direct-to-consumer product. In this case, it goes directly to physicians, mostly primary care physicians. So a whole another strategy to overcome these hurdles. And so after the Research Roundtable for Epilepsy discussion of these various issues and considerations, it's convened 3 working groups. The first one is what we're calling ground truths for seizures. So one thing to overcome for device companies is that the FDA would compare all new things to what we're calling ground truth. The FDA, I'm sure, would never call it that, but what we'd call ground truth. And so for seizure counting, the ground truth has been patient-reported outcome of seizures in a seizure diary. Well, we already heard how the seizure diary is entirely inaccurate today. So what if you have a device that works better than the diary? That's a really difficult thing to figure out. So that's an example of ground truth for seizures. There's also certain specific considerations about that, about how you decide something's a seizure on EEG, which is another conundrum for device developers. The second, I've spent too much time on reimbursement, but it's really the driver for this, and there's a work group about that. And then wearables. So although we tend to think of wearables, there's many other devices besides wearables that I didn't mention, those 27 companies doing device and technology development. And so wearables is just one subgroup that have their own considerations. And so there's a subgroup working on that. And I'd encourage any of you that are interested to ask us more about that if you have some other ideas or would like to contribute. So in summary, innovation is thriving in epilepsy for SUDEP prevention devices. We're just at the beginning, but innovation is all at the beginning. That's what makes it innovative. So just a lot of ideas. 27, as I mentioned. So I would have displayed them, but it changes every day. I'm sure we're up to 35 if we were to keep track in detail of different people developing these. The second important thing is that business development and reimbursement are unpleasant and complicated problems, but must be solved in order for us to get devices available to prevent SUDEP, undoubtedly. And that there are viable business development and regulatory pathways. I just mentioned them very superficially, but viable pathways exist. And so device developers must be supported to help them learn these viable methods. And that's really maybe the most important takeaway today, is that if we want to have devices available to prevent harm to people with epilepsy, we have to support the device developers in a different way if we support other kinds of development in epilepsy because of these unique challenges I've mentioned. So thank you.
Thank you. We'd like to have all the speakers come up for. We only have time for 2 questions, unfortunately, but I'm sure all of the speakers will be available after people have questions. So, yeah, over in the corner.
Really lovely talks. I learned so much during this session, really the whole day. This question is more directed to Heather and Luis. I'm not a clinician, so bear with me. It was great to see the update on the ASO works. With lumbar punctures, how practical is doing repeated punctures? And is there any, I guess with the therapies that are already out there, are there issues with compliance because of the procedure? I don't know how, I think from what I know, it shouldn't be like that painful, but I don't know about the repetitive nature of doing that for a disease.
So I don't actually know the data, that's a good question, but this is what's been done for spinal muscular atrophy, for example, for the ASO. It certainly can be done, and often kids need to be sedated somewhat, right, to make it effective and efficient delivery of the drug. But as far as I know, there haven't been issues with with it failing over time for some reason. And if anybody has any other data, I'd be happy to hear it. But I don't think that is particularly an issue. I think in general, because of the effectiveness, especially for something like spinal muscular atrophy, that families have been compliant. But I will also say that I think a lot of them are opting for the gene therapy. So there are multiple approaches for SMA. This is the one I'm most familiar with. But there's a gene therapy, so gene replacement. There's an ASO, there's a small molecule. So there are several things that have been developed. And I think, for those who qualify, I think the gene therapy is an option that many go with. So.
As far as I know, there are very few kids that being for Dravet syndrome ASO. Like 2 or 3 only have been taken out from the study because they don't come back for treatment, but there are very few.
Across the room. I appreciate it.
This question's for Nathan. So I'm part of a company called Cetera, and we're a device company. So everything that you've mentioned are exactly things that keep me up at night. But my question for you is particularly around clinical trials for SUDEP. As you had rightfully mentioned, although it's not rare, it's difficult to capture. So we're in a space where we're trying to prevent SUDEP, but based on the working groups that you've been a part of so far, have there been any thoughts on solutions on how to go about doing said clinical trials to demonstrate prevention of SUDEP in a somewhat practical way? Thanks.
Well, I guess the short answer is no. So the usual strategy is to find something else. And I guess that's what I was alluding to, that it is surmountable. So there's a whole process that has to do with drug and device development is that you'd like to find the Holy Grail, but it's unlikely you're going to find that. If you find that, then typically there's no hurdles at all because everything's so overwhelmingly successful. More often, you're trying to affect something that you think is going to affect SUDEP. And I think it's probably unlikely we're going to find something that's measurable because of the nature of just exactly what you said. So instead, reducing seizure frequency is a perfectly good outcome. For most people, they'd say, well, that's not bad. It's a reasonable outcome. It's what we use for drugs, regardless of whether it prevents SUDEP, although there's lots of inferential information that prevents SUDEP. So, in FDA, in clinical trials submitted to the FDA, the incidence of SUDEP is 6 times higher in the placebo arm than the effective drug-treated arm. So we already know that to be the case. So I think redesigning the way to think, and I think drug developers have been thinking this way for a long time, so they often have a secondary goal, but they know there's a primary outcome measure that's measurable, that is meaningful, that affects this other more important thing. And I think the same would be true of devices. So for instance, merely detecting a seizure is probably sufficient to start. And there's lots of reasons why that would be true for live ongoing seizure detection, somewhat for retrospective seizure detection, and then of course could lead to prediction.
We have time for one more question, if there is any from the audience. Otherwise, I have a question. We talked a lot about the overlap between SUDEP and sudden cardiac death, and the fact that a lot of these epilepsy genes are expressed both in the brain and the heart. And just wondering about your thoughts on regularly screening not just our genetic epilepsy patients, but our epilepsy patients with cardiac investigations, whether it be ECG or Holter or echo. With fenfluramine available for Dravet patients, we're doing more monitoring now. But whether this should be standard of care for our patients, whether there's any comments about that.
Yes, we have been suggesting that every patient has to be monitored also for EKG And we have a couple of cases where we learned, not working with animals, but iPSC-induced cardiomyocytes, that we found dysregulation of sodium currents. Then the patients were, they did some cardiac work and they found abnormalities. So now they are under treatment. So yeah, I think it should be something that has to be done every time.
So I'm just going to make a comment. In our community, we do recommend an EKG once a year, and it's to look for lung QT and some abnormalities. So,
I mean, I think it's, I don't have a formal recommendation, but it's a non-invasive, pretty easy screening to do. So, and it certainly wouldn't hurt.
Great. So unfortunately, we don't have a time for additional questions, but thank you so much to all of our speakers today. Really a wonderful session and very inspiring.
Yeah, do you all, hey, the panel was great. Do you all mind while we close up here? Thank you. Thank you all so much. That was a fantastic panel. I know we only have a few more minutes, so I hope you can bear with me. We want to end on a high note. We've got a few announcements. I've got 2 thank yous, and then I'm going to ask 2 folks to come on up, and we'll close out. But I just want everyone to please help me give a round of applause to the speakers today, who were, I thought, phenomenal. Thank you all so much. And then I think Eileen Murray is in the room, and if she could come up, we have a little something for her. She, as most of you probably know, is moving on from AES after being here for a very long time, and she has been a huge champion of PAME ever since the beginning, and it's really made all the difference to be a part of PAME, and she has fought for all of the support we've gotten. So we're so grateful. We're going to miss you dearly. One of the things we always talk about in the leadership group is how we see PAME as kind of like a convener of, all these different pieces. We help, hopefully, to try and put this puzzle together of what is a very complicated problem around epilepsy mortality. So Eileen, not that you're retiring, but in your quieter times, we got you a really fancy puzzle. Thank you, Eileen. And you are truly you. Oh, thank you so much.
Thank you.
We're just trying to be quick.
That's okay.
And then Gordon Buchanan, if you want to come on up here, he's going to help us close out on some exciting notes. And then George, do you want to make your way up here too while Gordon is doing his thing?
All right. Thank you, Gardner. So I'm here to present our abstract award winners. So we had, I think, like a couple hundred abstract submissions. And we had a great committee, Ruth Sanju, Cameron Metcalf, and Elena Laughlin, who did the heavy lifting of reviewing all the abstracts. And we have, I think, 50 that we chose to present here. And so we have 3 abstract award winners. So I don't know what order we're in here. I'm just going to, when I call your name, please come up, and I'll give you your certificate. All right. First is Hatif Purmatabed. I should have looked at these a little more closely. All right. Awesome. Congratulations. All right. Next we have Benjamin Kreitlow. I know him, yes. Congratulations. All right. And last but not least, we have Haley Pissick.
Thanks, Gordon. I mean, that's the best thing about this meeting are the younger researchers, because they provide all the hope for why we are here. And George came up to me last night at the welcome reception, and he said, Gardner, can I show some slides tonight? And I was like, well, George, I don't know if there's a projector at the reception, but maybe we can squeeze them in. And because George is ever so hopeful, he wanted to share some slides with you to, I think, end really on a positive note.
To answer some questions that keep coming up. So I want to show just a couple slides on funding, and but first I want to just thank all the speakers for their excellent talks, especially the bereaved, who all gave incredible talks that motivate all of us researchers. And I also particularly want to congratulate the program committee. I was on it for several years. I know how hard work it is to put together a good program, and this year has been really excellent. So thank you very much. So a lot of people worry about the health of the field of SUDEP research, and they bring it up to me a lot, asking, what do we think about the health of the field? And the quality of this program this year is a really great indication of how healthy the field really is. So I see a lot of positive for the future. Back in 2013, I made this graph and I added to it to update it. And other than a drop at the time of after COVID, which is probably a result of a decrease in the amount of research people could do during COVID, we've had a steady increase in publications. But still, people ask me about funding, and particularly about whether or not the NIH's investment in the Center Without Walls has decreased the amount of funding, or if it's been the same, or if it's increased. And so I should just give you a little history. I think that funding for SUDEP really started with CURE, pushing forward the idea that we needed to study this problem. And then AES really helped a lot to keep it, to help increase the growth. And then the NIH really stepped in and really pushed this forward, the funding for this, which brought a lot of new people into the field. And I'd like to thank Vicki Whitmore, who I think is here, who allowed me to be the first one to show this slide publicly. And you can see that at the time of the funding of the SUDEP Center Without Walls, or Center for SUDEP Research, that there was actually an increase in funding that occurred after 2 years of the funding for the Center Without Walls. So it certainly didn't slow down the growth, because that was additional on top of the Center Without Walls. And then after that, it has continued to increase NIH funding, and we've gone from $6 million per year to $18 million per year, which is an incredible growth over that time period. How do I switch? Oh, somebody else is doing that for me, I guess. But you can go to the next slide. And the total amount of NIH funding for SUDEP research has been $135 million, which it's expensive to do research, but that's a really great amount for the NIH to support. And if you go to the next slide, and most of it's coming from the NINDS, and that's shown here as well. And then you can finish up with the last slide. There's a lot of different areas of research that are relevant to SUDEP, and so they've really tried to increase their coverage of all different kinds of types of research. And I really think it's important for people to know about this, because we need to support the NIH. It really is a jewel in the crown of the government. It helps a lot of research, not just SUDEP, but we need to be sure to advocate for that, for the funding. So thank you very much.
You can sit up here with me. Okay, thanks, George, for that pop-up presentation. It's really great to see positive trends. So 2 things, and then I know I'll shut up. Okay, so the reception, right, is down there, and please go and learn about all that great research and have fun and meet people. Your drink ticket is your name badge, so you can use that to get a drink, and then if we can recycle these at the registration table, that would be wonderful. And then I think the last slide, is the evaluation. Please fill that out. Like I said, tell us what worked, what didn't work, and really what you want to hear next year. And then if we're still on time, I would love, we've done this in the past few years, we've passed the mic around for a few people who might just want to say, share what their greatest takeaway was of being here. It's a nice way to close out if anyone wants to share something that they learned today or is taking away. Not really wanting to put anyone on the spot, but sometimes people want to share. Back there.
Miss Maggie. Hi, my name is Maggie Calvert. I lost my sister, Molly Calvert, last May, May 8th, 2023. This is my second time at PAME, and I'm just so thankful to everyone for being here, especially the people who organized it. If you're a scientist, please keep doing the work that you're doing. It's so appreciated. If you're bereaved, please remember that you're not alone and that we're all here for you. My biggest takeaway was the legislative toolkit. I really, really appreciate all of the work that went into that. Thank you so much for your talk, Dominique. I am a public policy girly. I have an MPA, so I'm very excited to use that knowledge to help and to get to talk about my sister more and hopefully make a difference in people's lives. So thank you, everyone.
Thanks, Maggie. Okay. Well, I think everyone's ready to get a drink, read some posters. But thank you all so much, Maggie. That was a perfect way to close out, really. Thank you all for being here.